nuclear membrane, chromatin, centrosomes, nucleolus, chromatid, centromere, chromosome, spindle fibers, cell membrane. The cell cycle is divided into two different phases: interphase and mitotic phase. Interphase is split into three different parts: G1, synthesis, and G2. In G1, the cell expands in size. In synthesis, the genetic material of the cell replicates. And in G2, all other organelles in the cell replicate. That concludes interphase. Now to the mitotic phase. The mitotic phase is split into two different parts: mitosis and cytokinesis. Mitosis has 5 different phases, the first being prophase. In prophase, the nucleolus disappears. The chromatin coils up to form a chromosome with a centromere and 2 sister chromatids. And the centrosomes move to opposite poles of the cell, pushed by spindle fibers made from microtubules. Next is prometaphase. In prometaphase, the nuclear membrane disappears, and the spindle fibers connect to the kinetochore of the chromosome. But not that there are 2 different types of spindle fibers: kinetochore spindle fibers and non-kinetochore spindle fibers. Non-kinetochore spindle fibers do not connect to the kinetochore of the chromosome. They push the chromosome to the equator of the cell. Next is metaphase. In metaphase, the chromosome reaches the equator of the cell. Next is anaphase. In anaphase, the kinetochore is removed, and the daughter chromosomes move up the spindle fibers using motor proteins. This destroys the kinetochore spindle fiber. Next, the spindle fibers lengthen the cell by pushing against each other. Next is telophase. In telophase, the nuclear membranes come back, and as well as the nucleolus. Also, the daughter chromosomes uncoil to form 2 new chromatin. That concludes telophase. Next is cytokinesis, the final step of mitosis. In cytokinesis, the cells and organelles in the cells split. In animal cells, a cleavage furrow is formed, which pinches out the cell into 2 daughter cells. In animal cells, the Golgi apparatus forms a wall of vesicles, which creates a cell wall, which pinches off the cell into 2 different cells. Next, these cells will go into G1 of interphase and begin the cell cycle anew.